This video presentation is focused on the use of salivary leucocytes as non-invasive sample to assess DNA damage and repair in human biomonitoring studies using the COMET assay, and has been created by Natalia Fernández Bertólez, Vanessa Valdeiglesias, and Blanca Lafón from Universidade da Coruña, Instituto de Investigación Biomédica de A Coruña, and Fundación Novoa Santos in Spain. It was prepared in the framework of the European Partnership for the Assessment of the Risks from Chemicals, as part of the work conducted in Work Package 9.4, dedicated to training. It is structured in two main parts. The first one dedicated to DNA damage, and the second part dedicated to the repair of DNA damage. In the first part, we will make a brief introduction to the concept of genotoxicity and describe the methodology of the COMET assay, one of the most frequently used techniques to evaluate DNA damage. We will also talk about the biomatrices used for applying the COMET assay in human population studies, and explain the optimization of the salivary leucocytes to be used in the COMET assay to assess DNA damage. The second part will address the repair of DNA damage. We will make a short description of the main DNA repair mechanisms, describe the fundamentals of the challenge-comet assay as a functional assay for evaluating the DNA repair process, and detail the optimization of this methodology using salivary leucocytes as biological sample. Part 1. DNA damage. Introduction to the COMET assay. Genotoxicity is the term used to describe the property of those agents that can cause DNA or chromosomal damage, leading to genetic mutations, chromosomal fragmentation, or abnormal gene expression. Genotoxic agents include certain chemicals, physical agents such as ionizing radiations, and some biological agents like viruses. DNA damage refers to alterations in the chemical structure of DNA that can disrupt its normal functions. DNA is the molecule that carries genetic information, critical for the development, functioning, and reproduction of all living organisms. Damage to DNA can occur naturally through errors during the replication process, or as a result of exposure to external factors such as ultraviolet radiation, chemical pollutants, or environmental toxins. Common types of DNA damage include single and double-strand breaks, base modifications, and cross-linking. If left unrepaired, DNA damage can be fixed and impair the cell's ability to accurately replicate and transcribe genetic material, potentially leading to mutations and genomic instability. The importance of studying DNA damage and genotoxicity lies in their potential consequences for human health and the environment. Persistent DNA damage can disrupt normal cellular functions, trigger cell death, or lead to uncontrolled cell growth, a hallmark of cancer. Additionally, DNA damage in germ cells can cause heritable genetic mutations, affecting offspring and contributing to genetic disorders. Beyond human health, genotoxic agents can have environmental consequences by affecting the genetic stability of organisms in ecosystems, which may lead to biodiversity loss. Moreover, studying DNA damage and genotoxicity is fundamental for understanding disease mechanisms and aging processes. Accumulated DNA damage is strongly linked to age-related diseases, such as neurodegeneration, and plays an important role in various chronic diseases. This understanding also underpins cancer research, as genotoxic stress is a key factor in tumorigenesis. Advances in this field have enabled the development of diagnostic tools and therapies, such as targeted cancer treatments that exploit vulnerabilities in DNA repair pathways. Brief history review of the COMET assay. Many in vitro and in vivo tests for genotoxicity have been developed to detect DNA damage or its biological consequences in prokaryotic or eukaryotic cells, providing a wide range of endpoints. Before the development of the COMET assay methodology, DNA strand breaks were evaluated using techniques such as alkaline sucrose sedimentation, alkaline unwinding, and alkaline elution. All these techniques are based on the detection of discontinuities in the single strands of DNA by breaking the hydrogen bonds between bases of both strands using an alkaline medium. The single-cell GIL electrophoresis assay is a well-established, simple, fast, and sensitive method to measure DNA damage in individual cells, and it is extensively used in human population studies. This methodology is also commonly known as the COMET assay, since after its performance, cells with DNA damage have the appearance of this cosmic body, with an intensely bright head and a tail whose length and intensity are related to the number of DNA strand breaks it contains, while undamaged cells appear as intact nuclei, with a round shape and without a tail. The development of COMET assay has resulted from key contributions over the past decades. Let's see a brief chronology of COMET assay development. Breeder and Johansson in 1978 observed that following X-ray exposure, the nucleus of cells lysed in alkaline medium became diffuse in a dose-dependent manner. They quantified for the first time DNA damage in single cells by embedding them in an agarose gel prepared on top of a microscope slide and lysing them under mild alkaline conditions. After neutralizing, they stained the cells with acridine orange and quantified the damage extension by estimating the proportion of green fluorescence, indicative of intact double-stranded DNA, with regard to the red fluorescence, indicative of damaged single-stranded DNA. The single-cell GIL electrophoresis assay was firstly developed by Austin and Johansson in 1984 to detect radiation-induced DNA damage. They lysed the cells embedded in agarose and subjected them to electrophoresis under neutral pH conditions. They described the migration of DNA towards the anode as a result of the relaxation of the supercoiled loops caused by the strand breaks. This version of the COMET assay, however, detects only double strand breaks, which are characteristic effects of radiation and radiomagnetic agents. The alkaline version of the COMET assay was later introduced by Singh and colleagues in 1988. This is the most frequently used version of the assay and is able to detect, additionally, DNA single-strand breaks and alkali labile sites, such as baseless sugars or apurinic and apyrimidinic sites, called AP sites, that are transformed into single-strand breaks under alkaline conditions at pH higher than 13. Furthermore, this alkaline version is more sensitive to detect DNA damage induced by genotoxic agents, since most of them induce single-strand breaks in a proportion several orders of magnitude higher than double-strand breaks. In the decade of 90s, advances were achieved in understanding DNA movement during the electrophoretic process. For instance, Olive and colleagues in 1991 noted that in neutral medium, the release of DNA fragments in the GIL is caused exclusively by double-strand breaks, as DNA remains in a duplex form. Furthermore, various laboratories proposed refined protocols to improve the assay reproducibility, specificity, and sensitivity for DNA damage detection, and used it for new applications. One of the most exciting developments was the use of specific endonucleases to detect particular types of DNA lesions, such as oxidized DNA bases, among others, that was introduced by Collins and colleagues in 1994. These enzymes convert the specific DNA lesions into strand breaks so that they can be detected by the COMET assay. Already in the 21st century, additional improvements were introduced in the COMET assay methodology. Regarding automation and analysis, tools for image analysis and specialized software were introduced to measure the shape and size of the comet tails, thus enhancing the objectivity and precision of the assay. More recently, several high-throughput approaches have been developed, such as the Comet chip platform, the 12-gel Comet assay, or the use of gel bond films. Other technical modifications and integration of the assay with advanced molecular methodologies have been also developed. Combination with in situ hybridization, which enables simultaneous identification of DNA breaks or gene amplifications and DNA damage. Combination with immunostaining, that uses antibodies to provide detection of specific DNA lesions, such as cyclobutane pyrimidine dimers. The combination with the mu fluorescent staining of bromodioxyuridine incorporated by the cells allows discrimination between cells with induced DNA damage and cells in the S phase of the cell cycle, which are undergoing DNA replication and therefore contain a physiological level of DNA discontinuities or gaps, which are detected as DNA breaks in the COMET assay. And combination with methylation-sensitive restriction endonucleases to measure changes in the global DNA methylation patterns with EPI-COMET chip. This methodology is a crucial tool in genotoxicity assessment, widely used in numerous areas due to several advantages. High sensitivity. It can detect from over a few hundred to several thousand DNA breaks per diploid mammalian cell. Low number of cells are required to conduct the assay, even less than 20,000. It is applicable to many organisms and cell types, as long as a suspension of single cells can be obtained. DNA damage evaluation can be conducted in non-proliferating cells. It allows detection of cell populations with different levels of DNA damage. It is well established with a simple and fast protocol. It has high versatility and flexibility. The equipment necessary is simple and not expensive, and no radioactive isotopes are used. Therefore, it can be incorporated in almost every laboratory. It also presents high applicability to many different fields of study. Although its most common application is in in vitro and in vivo genotoxicity testing, to screen drugs, cosmetics, chemicals, etc., there are many scientific areas in which the COMET assay is used. For instance, it is widely used in human biomonitoring, including the study of occupational or environmental exposure to hazardous agents, nutritional studies, and to assess background levels of DNA damage. In environmental monitoring, and also in mechanistic toxicology to understand the mechanisms involved in DNA damage and repair. All these advantages motivated the fact that the COMET assay was validated by the European Centre for the Validation of Alternative Methods and that OECD approved a test guideline for the in vivo version of the assay in mammals. Methodology of the COMET assay. The alkaline version of the COMET assay is the most commonly used in many studies to assess DNA damage and DNA repair, and has widespread application in genotoxicity testing both in vitro and in vivo, since under alkaline conditions, the assay detects overt strand breaks, which can include single and double strand breaks, as well as transient excision repair induced breaks. It also detects lesions that are alkali labile, which include apurinic and apyrimidinic sites, also called AB6 sites or AP sites, And moreover, since virtually any cell population or single cell suspension from any tissue type can be used for analysis, the assay is widely used in environmental biomonitoring and in human population monitoring. The first step for conducting the assay is the isolation of cells from whole organisms, animal or plant tissues, biopsies, blood samples, spheroids, or cell cultures. Blood cells are the most convenient in human biomonitoring studies, as they are already a single-cell suspension. Likewise, cells growing in suspension cultures can be used directly in the COMET assay, whereas adherent cells must be detached from the surface of the culture plate and resuspended in a suitable medium. Cells from spheroids, tissues, biopsies, or whole organisms need to be dispersed and homogenized before processing for the COMET assay. The methodology of the alkaline COMET assay is as follows. The cell suspension is embedded in low melting point agarics, and a microgel is prepared on a microscope slide pre-coated with normal melting point agarics. Then, cells are lysed by a solution containing detergents and high salt concentration in order to release the nuclear content in the form of nucleoids. Afterwards, the slides are incubated at strong alkaline conditions, pH higher than 13, to achieve DNA relaxation and unwinding. During electrophoresis at the same alkaline conditions, DNA fragments migrate to the anode due to their negative charge. Subsequently, slides are washed with a neutralizing solution and stained with a fluorescence DNA binding dye. Thus, sensitivity of the alkaline COMET assay is greatly influenced by the pH of the lysis and electrophoresis buffer. Data on DNA migration from each individual nucleoid are collected under fluorescence microscopy, either by visual scoring or by using computer-automated image analysis systems. The COMET descriptors used by image analysis systems are tail length, percentage of DNA in the COMET tail, and tail moment, which is the product of tail length and tail DNA. There has been debate about the suitability of the different COMET parameters, but the current belief is that the percentage of DNA in the COMET tail is the best COMET descriptor, based on its linearity at a wide range of DNA damage. In case a computer image analysis system is not available, visual scoring can be performed to evaluate COMET assay results. Visual scoring uses a semi-quantitative scale that categorizes comets into different classes on the basis of how they look in the microscope. The sum of scores is reported as arbitrary units, which depend on the number of comet classes and the number of comets scored. A recent paper assessed the intra and inter-investigator variability in visual scoring of comets. The results obtained demonstrate that visual scoring is a reliable way of analyzing DNA migration in comets, although further standardization of visual scoring is desirable. Usually, a minimum of 100 comets per experimental point, 50 per replicate, are scored. Enzyme-modified COMET assay. More precise information on the nature of the lesions detected in the DNA can be obtained by the inclusion in the assay protocol of lesion-specific endonucleases, which convert some types of DNA damage into strand breaks. For example, the incubation of the nucleoids after lysis with glycosylases, such as from amidopyrimidine DNA glycosylase or FPG, has been used to detect with high sensitivity oxidative DNA damage, principally 8-oxoguanide, although it also detects some types of alkylation damage. 8-oxoguanine is the most frequent DNA lesion produced in the presence of oxidative stress. Therefore, it is considered as a good biomarker of oxidative DNA damage. Other enzymes used for this purpose are the FPG human homologue 8-oxoguanine DNA glycosylase 1, HOGG1, that is a more specific for oxidation products and specifically detects 8-oxoguanine, and endonuclease 3, or ENDO3, that particularly recognizes oxidized pyrimidines. For conducting this version of the COMET assay, after obtaining the cellular suspension, duplicate slides with 2 agarose drops per slide are prepared for each experimental condition. After the lysis step, slides are washed 3 times for 5 minutes each with enzyme buffer, and then slides are split into 2 groups and treated with either enzyme or just enzyme buffer, covered with cover slips and incubated at 37 degrees in a humidified box. After this incubation, slides are processed as in the alkaline COMET assay. Enzyme-modified COMET assay can also be conducted with higher throughput versions of the COMET assay, namely the COMT platform, the 12-GIL COMET assay, or the use of GIL-bound films. The concentration at which the enzyme is used and the length of the incubation period is critically important. It should obviously be high enough to detect all the lesions present, but not so high that non-specific damage is detected. Hence, titration experiments are necessary with each enzyme batch, using cells that are known to contain the lesion of interest, ideally cells treated with an appropriate agent. A plateau should be reached at certain enzyme concentration and incubation time, reflecting optimal detection of lesions. Then, slides are analyzed by fluorescence microscopy and comets scored with an appropriate software. Net enzyme-sensitive sites are calculated by subtracting the percentage of DNA in the comet tail obtained after buffer incubation from that obtained after the enzyme incubation for each experimental condition. What is the principle of the COMET assay? The COMET assay is based on the principle that the strand breaks reduce the size of the large duplex DNA molecule, and under strong alkaline conditions, relaxed strands are cooled out during electrophoresis. The high salt concentration in the lysis step of the assay removes the cell membranes, histone proteins, cytoplasm, and nucleoplasm, and disrupts the nucleosomes, leaving behind the nucleoid consisting of the negatively supercoiled DNA. The breaks present in the DNA cause local relaxation of the supercoils, and loops of DNA are then free. The high pH, higher than 13, in the unwinding step facilitates the denaturation of DNA, with disruption of hydrogen bonds between the double-stranded DNA, and expression of alkali labile sites as frank breaks. During electrophoresis, this damaged DNA migrates towards the anode, thus forming the distinct comet with a head containing the intact DNA and the tail containing the damaged DNA that can be visualized after fluorescence staining. In the enzyme-modified COMET assay, cells are incubated with the lesion-specific endonuclease after the lysis step, so it is applied directly to the nucleoids embedded in the agarose. During the incubation period, this enzyme induces extra breaks where the specific lesions it recognizes are present, and these breaks are measured by continuing with the standard assay protocol. As a control, duplicate nucleoids are incubated with enzyme buffer, but without enzyme. Biomatrices used for COMET assay in population studies. As already mentioned, in the standard alkali version of the COMET assay, it is possible to detect strand breaks, both single and double, alkali labile sites, and incomplete excision repair sites. And the versatility of this assay allows the combination with different repair enzymes, making it possible to detect specific types of DNA lesions. The enzyme HOGG1 recognizes 8-oxoguanine, the most frequent lesion produced in the presence of oxidative stress. Therefore, it is used to specifically detect oxidative DNA damage. Peripheral blood leukocytes, isolated from whole blood, are the most common cell type used for this assay in human population studies. So they are considered the gold standard sample for this assay. Human peripheral blood is a very convenient source of cells because it is easily obtained, and cells are available in large numbers. Leukocytes circulate throughout the whole body, and thus can be seen as reflecting the overall state of the organism regarding exposure to genotoxic agents. The COMET assay is ideally suited for use with cells such as peripheral blood leukocytes, which are already in suspension. The advantages of using peripheral blood leukocytes as biological sample in the COMET assay include the following ones. Blood sampling is a minimally invasive procedure, making it relatively easy and acceptable for both clinical and research settings. Peripheral blood leukocytes are readily available in sufficient quantities for the COMET assay, avoiding the need for complex isolation techniques. As part of the circulatory system, leukocytes provide a good representation of systemic exposure to genotoxic agents. Blood collection and leukocyte isolation are well-established and standardized procedures, ensuring consistency across studies. Isolated leukocytes can be cryopreserved for later use, enabling longitudinal studies or comparisons across multiple time points. Leukocytes are highly sensitive to DNA damage, making them a reliable cell type for detecting genotoxicity. Peripheral blood sampling is particularly suitable for human biomonitoring and epidemiological studies, allowing researchers to assess DNA damage in a non-invasive way. Peripheral blood leukocytes are widely used in clinical studies to monitor DNA damage in health and disease, making them a valuable tool for diagnostic and therapeutic evaluations. It is a standard practice in genotoxicity testing to keep records of results of tests of positive and negative controls as a historical control dataset. This is important for quality control, defining an acceptable level of variation, and assessing assay reliability. In the case of human biomonitoring, it is also important to build a database of background damage levels in peripheral leukocytes from control populations. This allows the consistency of the assay to be monitored over time. However, collection of whole blood samples involves an invasive procedure and requires participation of healthcare professionals. Moreover, vein puncture could be problematic in some population sectors, such as children, patients with dementia, or subjects with vein problems, such as older adults or cancer patients subjected to chemotherapy. Furthermore, the large volume of blood required for leukocyte isolation not only discourages collaborations in multidisciplinary research projects and limits the number of biomarkers that can be analyzed in the sample volume obtained, but also increases the probability that the subjects refuse to participate in the study. For all these reasons, different cell types and tissues have been proposed as alternatives to the use of blood leukocytes to assess DNA damage. Among these alternative biomatrices, samples from the buccal cavity, and in particular salivary leukocytes, present several advantages, including that they can be collected by a non-invasive procedure and participants may even collect the samples themselves. The processing of these samples is quite simple, and they can provide interesting information on exposures by inhalation or by ingestion. Nevertheless, a review by the challenge-comet group pointed out some problems regarding the use of salivary leukocytes, such as the low number of cells retrieved per sample and the interference caused by the buccal epithelial cells that makes the microscope scoring more challenging and time-consuming. Besides, this approach has not been standardized yet, and there are different aspects of this approach that remain to be addressed, such as its suitability to detect different types of DNA damage, both primary and oxidative, the possibility of using cryopreserved cells, very convenient for biomonitoring studies, or the comparison with the sensitivity obtained when employing the gold standard, peripheral blood leukocytes. In the first section of this video presentation, we will also show the process of standardization of the COMET assay to detect DNA damage using salivary leukocytes as biomatrix, both in fresh and cryopreserved. The objectives of the standardization process were to test the validity of salivary leukocytes as a proper biomatrix for the COMET assay, to evaluate the ability of this approach to detect different types of primary and oxidative DNA damage, and to determine whether frozen salivary leukocytes are still suitable for displaying those types of DNA damage. To that aim, leukocytes isolated from saliva samples were obtained from 6 healthy non-smoking volunteers, 3 men and 3 women, by performing 4 consecutive mouth rinses with 10 milliliters of saline solution. After centrifuging and washing, the 6 rinses were pulled together, and leukocytes were isolated by density gradient centrifugation using histobake. One fraction of pulled leukocytes isolated from saliva was used in fresh, and the other one was resuspended in freezing medium, containing 10% of dimethyl sulfoxide as cryopreservative and frozen at -80 degrees for at least 1 week. In order to compare results obtained from saliva leukocytes with the gold standard, the blood leukocytes, whole blood samples were obtained from the same volunteers by vein puncture, using percutaneous CPT tubes, and the leukocytes were isolated following the manufacturer's instructions. Isolated blood leukocytes from all donors were also pulled together, resuspended in the same freezing medium, and stored frozen until analysis, at least 1 week. The fresh saliva leukocytes and the 2 types of frozen leukocytes, after thawing, were treated in vitro with 4 DNA-damaging agents. 3 well-known genotoxic agents were employed to address the suitability of fresh and frozen salivary leukocytes to respond to different types of primary DNA damage and be detected by means of the standard COMET assay. Specifically, cells were treated with methylmethane sulfonate, an alkylating agent that methylates DNA bases, essentially guanine, and, to a lesser extent, adenine. These lesions are considered as a source of DNA double-strand breaks as a result of collapsed replication forks at the lesions or processed intermediates. Cells were also treated with the intercalating agent actinomycin D, an antitumor drug that particularly intercalates into transcriptionally active regions of chromosomal DNA and thereby avoids RNA synthesis, leading to formation of DNA single and double-strand breaks. And cells were exposed to ultraviolet radiation, which is a cross-linker. In particular, UV radiation induces cyclobutane pyrimidine dimers and 6,4 photoproducts in DNA, which are repaired by nucleotide excision repair pathway. In order to detect oxidative DNA damage, cells were treated with potassium bromate, which causes oxidized guanines but do not induce strand breaks. Hence, potassium bromate is a suitable positive control for the HOGG1 modified COMET assay that detects the oxidized guanines and introduces a DNA break at the site of the lesions. Also, leukocytes were treated with distilled water, which was used as negative control. All treatments were performed in triplicate, and the doses used of the 4 genotoxic agents, as well as the treatment times, were selected on the basis of previous studies. Immediately after treatments, and to assess primary DNA damage, cells treated with methylmethanosulfonate, actinomycin G, and UV light were mixed with agarose, and the alkaline COMET assay was performed following the standard protocol explained before. Complementarily, after potassium bromate exposure, the HOGG1 modified COMET assay was conducted to evaluate the oxidative DNA damage induced. To that aim, an incubation step with a glycosylase at 37 degrees for 30 minutes was carried out. At the end, slides were analyzed by fluorescence microscopy to determine the DNA migration in each condition, using the percentage of DNA in the tail as COMET descriptor. When evaluating genotoxicity, it is recommended that the treatments applied do not exceed 20% of cytotoxicity, to avoid false positive results due to cell death not related to genotoxicity. Therefore, in these experiments, cell viability was checked after treatments by trypan-blue exclusion technique, resulting higher than 80% in all cases. Results obtained in the standard alkaline COMET assay. In all cases, fresh and frozen salivary leukocytes and blood leukocytes, cells exposed to the 3 agents inducing primary DNA damage showed significant dose-dependent increases in DNA damage, regardless of the cell sample used, demonstrating the sensitivity of all of them to the induction of genetic damage of different origin and their suitability for the COMET assay procedure. Furthermore, the response of the salivary leukocytes was quite similar to the response of blood leukocytes, and consistent with several previous studies that evaluated induction of DNA damage by the same genotoxic agents on different types of non-leukocyte cells. Similarly, the results obtained from the 3 cell samples analyzed after exposure to potassium bromate showed progressive increases in the net HOGG1 sensitive sites. The dose-response relationships were significant in all cases. These results, together with the ones obtained in the standard alkaline COMET assay, confirmed that there is no influence of the cryopreservation process on the response of salivary leukocytes to the induction of DNA damage, both primary and oxidative. Furthermore, the process of leukocyte isolation from saliva samples resulted very convenient, since it made it possible to avoid interference in microscopic images caused by exfoliated cells from the buccal mucosa, described in previous studies. Thus, the scoring process with isolated leukocytes is simpler and faster. When comparing the baseline level of primary DNA damage in the 3 different sun samples, no difference were observed between fresh salivary leukocytes and blood leukocytes, and frozen salivary cells showed only a slight significant increase in the baseline genetic damage relative to the other biomatrices. In line with these results, Russo and colleagues obtained a similar baseline level of primary DNA damage from saliva and blood leukocytes, both analyzed in fresh. Regarding the basal level of oxidative DNA damage, no differences were observed between the 3 biomatrices. Supporting this observation, a review by the H-COMET group concluded that various types of blood samples can be cryopreserved with only a minor effect on the basal level of DNA damage, both primary and oxidative. However, Marino and colleagues pointed out that basal levels of damage could be influenced by the cryopreservation time, becoming significant after long periods of cryopreservation, from 6 months on, which were not tested in the current study, since these samples were kept in the freezer for no longer than 1 month. Finally, it is worth noting that the yield of leukocytes in the process of isolation from saliva samples is relatively low, approximately 200,000 cells per donor. However, the purity and cell viability of the cells isolated is high, so the number of cells obtained is more than enough to perform the COMET assay for human population studies, assessing both primary and oxidative DNA damage. On the basis of the results obtained in the standardization process, the following conclusions can be drawn. Salivary leukocytes have been optimized and validated as a suitable biomatrix for the COMET assay. They have been demonstrated to be sensitive to detect different kinds of DNA damage, both primary and oxidative, by the COMET assay. And frozen salivary leukocytes are suitable alternative for COMET assay biomonitoring studies, with similar sensitivity than blood leukocytes. For further details on the standardization process, you can consult this publication. Section 2. Repair of DNA damage. Cells' ability to detect and repair DNA damage is essential to maintaining the structural integrity of its genome in general and its genes in particular. Analysis of DNA repairability in human populations is an attractive biomarker for clinical investigators because alterations in several DNA repair pathways are linked to both heritable diseases and sporadically occurring age-associated diseases, such as neurodegenerative disorders and cancer. And it has been recommended that molecular epidemiological studies include the assessment of individuals' ability to remove DNA damage. Throughout evolution, the cell has acquired a series of mechanisms that tend to reduce the damage that occurs in DNA, all of them aimed at maintaining genomic stability. The main characteristics of repair systems are their ubiquity, high complexity, redundancy, efficiency, interspecific homology, and their functional variability. DNA repair pathways. Human cells employ several distinct DNA damage repair pathways to maintain genomic integrity. Here's an overview of the main types. Base excision repair deals with small non-helix distorting base lesions, such as those caused by oxidation, alkylation, or deamination. It consists of the excision of the damaged base by a DNA glycosylase, leaving an abasic site. This is followed by strand cleavage, gap filling by DNA polymerase, and ligation. Nucleotide excision repair corrects bulky helix-distorting lesions, such as thymine dimers from UV light or chemical adducts. In its repair, a short single-stranded DNA segment containing the lesion is removed, followed by DNA synthesis and ligation. The repair of double-strand breaks can be performed by two different pathways: homologous recombination or non-homologous end joining. Homologous recombination repairs double-strand breaks using a homologous DNA sequence as a template. Hence, it uses the sister chromatid for error-free repair, making it most active during the S and G2 phases of the cell cycle. Non-homologous end joining repairs double-strand breaks by directly ligating the broken ends, without a homologous template. This process is quick but error-prone, as it may lead to small insertions or deletions at the repair site. It is active throughout the cell cycle. Mismatch repair fixes replication errors, like base mismatches and insertion or deletion loops that escape proofreading by DNA polymerases. This process consists of the recognition and excision of the mismatched segment, and then the DNA is re-synthesized. Direct repair directly reverses specific types of damage without removing the affected base or nucleotide. For example, the enzyme O6-methylguanine DNA methyltransferase repairs alkylated guanine by transferring the alkyl group to itself. And translation synthesis allows replication to proceed past a lesion when the normal DNA polymerase is stalled. It involves specialized DNA polymerases that can synthesize DNA across damaged templates, often introducing mutations. Each pathway is critical for a specific type of DNA damage and contributes to protecting the cell from genomic instability, which can lead to cancer, aging, or other diseases. Individual differences in DNA repair in human or animal studies can be analyzed by different approaches. Analyzing polymorphisms in DNA repair genes to determine how they impact repair efficiency, and determining changes in the expression levels of DNA repair genes by studying the levels of mRNA and proteins produced. This can be indicative of how cells respond to DNA damage, since low expression may suggest impaired repair capacity, while high expression could mean robust repair or compensatory responses. However, the functional effects for many polymorphisms is still unclear, and these methods do not consider post-transcriptional or epigenetic modifications. In addition, numerous genes are involved in the different DNA repair pathways, so determining the effect of a single one might be challenging, and they also interact with one another and may have overlapping specificities. Challenge-comet assay as functional assay. In contrast to the methods just mentioned, phenotypic or functional DNA repair assays provide a more complete picture of the whole DNA repair process, since it depends on the restoration of the normal DNA structure, also considering the possible influence of environmental factors. In this scenario, the challenge-comet assay is a functional biomarker of DNA repair, and is a simple approach, especially useful in human biomonitoring studies. It is based on the induction of a certain quantity of DNA damage in a cell sample, using a known genotoxic challenge agent. And after a certain repair period in the absence of the challenge agent, the remaining damage is evaluated by means of a genotoxicity test. Originally, cytogenetic assays were used for genotoxicity assessment, initially chromosome aberrations, and afterwards the micronucleus test. Later on, the COMET assay was used in combination with the challenge assay, providing the challenge-comet assay with all the advantages of the COMET assay, namely sensitivity, versatility, economy, need of a relatively small number of cells, and no requirement of ex vivo cell culture. A further advantage of the challenge-comet assay is that repair kinetics can be followed up at consecutive time points. The most straightforward approach to measure DNA repair activity is to induce DNA damage in cells and subsequently monitor the rate of removal of these lesions over time. Interestingly, the COMET assay was developed to measure DNA repair from the very beginning, to follow the reduction in the number of radiation-induced DNA breaks with time, which represents the repair of those lesions. Therefore, Singh and colleagues used what is now referred to as challenge assay to follow the kinetics of removal of DNA lesions and religation of the remaining strand breaks with time. The challenge-comet assay has been widely used for measuring the repair activity of cells and tissues. With the challenge-comet assay, it is possible to evaluate capacities in a variety of DNA repair pathways, or even in different steps of the same pathway, by employing specific genotoxicants as challenge agents or different DNA repair inhibitors. The traditional version of this assay was based on the application of X-rays as the challenge agent and the use of peripheral blood leucocytes as cell sample. Recently, it has been demonstrated the utility of employing other different challengers, namely bleomycin, methylmethane sulfonate, and UV radiation, thus enabling evaluation of alterations in different DNA repair pathways on the basis of the different types of DNA damage induced. Moreover, challenge-comet assay has been standardized for the use of whole blood samples, both fresh and cryopreserved, which are much more convenient in the case of biomonitoring studies. However, as mentioned before, obtaining blood samples is not feasible in all cases, especially in certain more sensitive populations. Since the use of saliva leukocytes for the COMET assay was optimized in the first section of this presentation, the question arises on whether they can be also valid for the challenge-comet assay. The final part of this video presentation addresses the standardization of the use of salivary leukocytes for the challenge-comet assay. And the objective raised was the validation of these samples, both fresh and cryopreserved, for assessing DNA repairability by means of the challenge-comet assay. Saliva samples were obtained from 10 donors, 6 women and 4 men, and leukocytes were isolated according to the procedure previously explained. These leukocytes were used either in fresh or after a process of cryopreservation for approximately 5 months. Before the challenge-comet assay procedure, these cells were incubated for 24 hours in the presence of fetal agglutinin in order to stimulate the cell cycle, since leukocytes are usually quiescent cells. They are in G0 phase of the cell cycle. After this incubation, cells were subjected to the challenge treatments with genotoxic agents inducing different types of DNA damage, namely bleomycin, methylmethanosulfonate, and UV light. 3 independent experiments were conducted, using 2 different doses for each challenge agent. Then, the standard alkaline COMET assay was conducted either immediately after the treatments or after 3 different repair periods in fresh medium, in order to study repair kinetics. Additionally, to minimize interassay variability, assay controls were introduced in each electrophoresis run, which were glial cells treated with methylmethanosulfonate. Cell viability was checked after treatments by means of propidium iodide exclusion, evaluated by flow cytometry, in order to avoid false positive results due to cytotoxicity. When cells are alive, their cell membrane is intact and the propidium iodide cannot enter. But when the cells are dying, the permeability of their membrane is altered, so they are positive for staining with a fluorescence dye. Cell viability in the study was in all cases higher than 80%, and in most cases higher than 90%, confirming the absence of cytotoxicity. This figure shows the results obtained with bleomycin treatments in salivary leukocytes. Both concentrations tested induced a significant increase in DNA damage over the control at time 0, notably higher in frozen leukocytes. The damage induced decreased with time in a linear way, returning to control values at time 3, 120 minutes, in all cases. These are the results obtained in a blood leukocytes in a previous study where cells were treated in the same conditions, which are very similar to those in saliva leukocytes, although DNA damage was not completely repaired at 2 hours. These data correspond to methylmethanosulfonate treatments. Again, significant increases in the tail DNA were induced at time 0, more notable in frozen saliva leukocytes. DNA damage values dropped gradually with time, following linear kinetics. At the end of the repair period, 2 hours, damage values indicated total repair, since they returned to control values. Results for blood leukocytes were similar, but in this case, total repair was not reached, although the longest repair time tested was only 60 minutes instead of 120. Results obtained with UV light exposed cells were similar in fresh and frozen saliva leukocytes. In this case, and as expected according to previous studies, the maximum level of DNA damage was obtained 1 hour after exposure, since this time is necessary for the maximal expression of strand breaks after exposure, mainly due to the nucleotide excision repair process. Afterwards, continuous descent in DNA damage was observed over time, according to a time-response linear kinetics. Complete repair for both doses was achieved at 180 minutes. These results were very similar to those obtained for blood leukocytes, and in this case, complete repair was also obtained at the end of the incubation period. Correlation analysis showed significant and very good associations between fresh and frozen saliva leukocytes, and also between the two types of saliva cells and blood leukocytes. It is worth mentioning that although the yield of leukocytes in the process of isolation from saliva samples is not high, the number of cells retrieved per individual is enough to conduct the assay using one particular challenge treatment and 4 time points, preparing 4 slides with duplicate drops per individual. In case of using other versions of the challenge-comet assay with a higher throughput, such as the Gilbon films of the 12-GIL comet assay unit, further experimental conditions could be assessed. In the analysis of the basal levels of DNA damage obtained at all time points evaluated, values were higher in frozen saliva leukocytes than in fresh cells, significant for some time points. But the magnitude of the difference was really low, less than 1.5% units in all cases, demonstrating that the cryopreservation does not induce additional DNA damage in these cells. Repair capacity at the end of the repair period was calculated according to the traditional formula reported in the literature, which considers the damage induced by the challenge agent before repair and after repair at the longest repair period tested. The traditional parameter was significantly higher in frozen than in fresh salivary leukocytes, due to the fact that the DNA damage initially induced was more pronounced in frozen cells, except in the case of UV light exposure. However, although total repair was observed in all cases in saliva leukocytes, values of repair capacity do not represent total repair. They are not close to 100. Thus, this parameter does not clearly reflect the actual repair capacity. Therefore, a new parameter was proposed for estimating the repair capacity, the net repair capacity, which considers the level of DNA damage in the control, basal level, when calculating the induced damage before repair and after repair. In this case, values of net repair capacity were pretty close to 100 in all cases, reflecting total repair of the damage induced. And no significant differences were observed between fresh and frozen cells, indicating that both biological samples were equally efficient in repairing DNA damage, regardless of their origin. Since several time points were evaluated, it was also possible to calculate the half-life, which is the time necessary for repairing half of the DNA damage induced. The results showed that the half-life was shorter in frozen than in fresh saliva leukocytes treated with bleomycin and methylmethane sulfonate, indicating faster DNA repair in frozen cells and very similar in cells exposed to UV radiation. The half-life provides information on the velocity of the repair process, and the net repair capacity reflects the efficacy of the repair process, namely the fraction of damage induced that could be repaired at the end of the repair period. Both parameters are independent of the initial amount of damage induced, and therefore are not influenced by the individual susceptibility to the challenge agent applied. Bleomycin is a radiomimetic compound that induces a wide range of mutagenic lesions, which eventually result in DNA single and double-strand breaks. Methylmethanosulfonate produces alkylation lesions and also causes double-strand breaks as a result of collapsed replication forks at the lesions or processed intermediates. And UV light is a cross-linker that causes covalent linkages between two adjacent pyrimidines. So, how are all these different types of lesions repaired? Single-strand breaks and alkylation lesions are mainly repaired by base excision repair, which takes a short time, usually less than half an hour. Repair of double-strand breaks by homologous recombination or non-homologous end joining requires a longer period, up to 120 minutes in this study. And pyrimidine dimers are repaired by nucleotide excision repair, a relatively slow process that in this case took 3 hours to be completed. On the basis of these considerations and the results obtained in the standardization of the challenge-comet assay in saliva leukocytes, the following conditions to perform the assay can be recommended according to the type of DNA repair to be assessed. For double-strand break repair, 20 microgram ml of bleomycin as challenge agent, with evaluations up to 120 minutes. For base excision repair, 75 microgram ml of methylmethane sulfonate, with evaluations up to 30 minutes for not capturing double-strand break repair. And for nucleotide excision repair, 300 microjoules per square centimeter of UV radiation, with evaluations from 60 up to 180 minutes. Based on the results obtained in this standardization process, the following conclusions can be drawn. Salivary leukocytes, both fresh and cryopreserved, for at least 5 months can be successfully applied in the challenge-comet assay, similarly to blood leukocytes. This approach is able to detect repair activity of diverse types of DNA damage, which indicate different DNA repair pathways. This approach is promising and especially useful for those populations for which collecting whole blood samples is difficult or not possible, and also when blood samples need to be used for the determination of multiple biomarkers. Moreover, the possibility of using cryopreserved samples facilitates the logistics of large human biomonitoring studies. For further details on the standardization process, you can consult this open access publication. Finally, these are the most important messages from this video presentation. The COMET assay is a rapid, simple, and sensitive method to evaluate DNA damage in single cells. Variations of this assay allow the evaluation of different types of DNA lesions. The challenge-comet assay is a functional assay to evaluate DNA repair activity and repair kinetics. Saliva samples are obtained non-invasively and can be suitable substitutes of whole blood samples in certain sensitive populations. Salivary leucocytes, both fresh and cryopreserved, are a suitable biomarkers for detecting different types of DNA damage by the COMET assay and can be also successfully applied in the challenge-comet assay to detect repair activity of diverse types of DNA damage. And these approaches are especially useful in human biomonitoring studies. Here you can see the references mentioned in this video presentation. This slide shows the affiliations and contacts of the authors and the Creative Commons license characteristics.